Welcome to Mr. Graham's Cell Parts, Eukaryotic Structure, Section 2. We have 9 cell parts to get through, so let's get started. The first one, the mitochondria. This is where cell respiration occurs. This is the little battery packs inside the cells, a source of energy for a cell. All eukaryotic cells have mitochondria, plants, animals, everything, because even though plants do photosynthesis, they still have to use that sugar later to make ATP, and they do that in the mitochondria. Mitochondria looks like a little bean with a bunch of folds in it. Here's a picture of a mitochondria, bean with a bunch of folds in it. That's exactly what it looks like. And there will be many mitochondria throughout a cell. The more energy the cell needs, the more mitochondria it would have. The second cell part is ribosomes. Ribosomes are the protein factories inside of a cell. Messenger RNA and transfer RNA would come together and they would bring amino acids in and link together at the ribosome. Ribosomes can be found free-floating out inside the cytoplasm, but many of them are attached to rough endoplasmic reticulum. That brings us to our next one, the endoplasmic reticulum, also shortened up to be called the ER because it's a lot easier to say ER than endoplasmic reticulum. The function of the ER is to transport materials. It's like a highway system that runs throughout the cell. Anything that needs to get moved around can get moved on the ER. Proteins, for instance, that get made on the ER then get transported throughout the ER. The other main function is to assemble lipid molecules. Any lipids that are needed for the cell membrane, phospholipids, for instance, get assembled on the rough or the smooth ER. There's 2 types, rough ER. Now, all eukaryotic cells are going to have ER. The ER sort of attaches to the nucleus and then weaves its way throughout the cell. Rough ER gets its name because it's covered in ribosomes, which makes little bumps all over it, which if you were driving a car around on it, it would be rough. Once those ribosomes would make the proteins on the ER, it would kind of insert them into the ER, and then they would get packaged up into a little vesicle, little pouch inside the ER, and transported throughout it that way. If anything that needs to be transported in the smooth ER, same thing. It's going to transport it wherever it needs to go into the cell. The smooth ER does not have any ribosomes on it, so therefore it's much smoother. Here's a picture of an electron microscope of a cell. Here you can see the mitochondria we looked at before. This is the nucleus we looked at before, and this is all ER. Picture an onion. If you slice an onion in half and you can see all of the layers of that onion, they are three-dimensional and they wrap all the way around the onion, but you're only seeing sort of like an alleyway of it, and that's basically what you're seeing here. ER is three-dimensional and it goes all throughout the cell, attaches to the nucleus, and then anything that gets made on it gets transported via a vesicle throughout the cell. A lot of times it will transport things to the Golgi apparatus. This is like the UPS of the cell. Once those proteins get to the Golgi, they get modified. The proteins might get changed and altered and sorted out, and they get packaged up into a new vesicle. And then that vesicle can either get stored in the cell, used in the cell, or transported to the cell membrane and then get secreted outside of the cell. All eukaryotic cells, again, since they all have ER, they're all going to have Golgi because they all make proteins. The Golgi apparatus looks like somebody sat on a stack of pancakes. Doesn't sound very scientific, but that's just what they look like. Here's one here, here's one down here, and here's one here. It's kind of compressed. It's narrow. It looks just like somebody sat on a stack of pancakes. And what happens here, these are compressed membranes, and in those membranes the proteins are getting sorted and packaged up into a vesicle here that gets created from one of those membranes. It's called a bleb. Here's another one down here. There's a bleb being made off of this Golgi apparatus, and this one is just in the process of starting to make one. Here's a smaller Golgi over here. Okay, this would all be endoplasmic reticulum, and you can see the little dots on it here. Those would be the ribosomes. Lysosomes are specialized vesicles that contain digestive enzymes inside the cell. They are used really to clean up the cell. If there's a broken part that doesn't work anymore, the lysosome would engulf it, take it in, and then digest it. If it's a protist cell that eats something else and stores it in a food vacuole. Eventually that food vacuole would fuse with the lysosome, and the digestive enzymes would break down and recycle those parts. Some lysosomes actually will sort of break apart and release digestive enzymes into the cell if the cell is near the end of its life, and it would actually destroy this cell from the inside out. Animal cells and protist cells are going to have lysosomes. You really won't find them much in plant cells. Cilia and flagella are two specific structures that are used to move cells around or to move things around the cell. Cilia are lots and lots of little tiny hair-like structures, and flagella is like a long tail-like structure, and there's usually very few, only one or just a couple, where cilia would be many of them all over the place. And either they use to kind of swish water and fluids around the cell to move things into the cell, or they are used to push the cell around or pull the cell around where it needs to go. So protists are going to have them, and there are certain animal cells that have them. Here's an example of stentor. We looked at that before. Well, these are cilia lining its mouth parts, and they create this swirling vortex of terror when little tiny microscopic organisms get sucked into this vortex and down into its mouth parts to get digested. These cilia might also help propel the stentor throughout the liquid and help it swim around. Paramecium, which we looked at earlier, are completely covered in cilia all around its body, so they can move around like bumper cars. A flagellum is more like a tail, so a sperm cell that has a single tail has a flagellum. That's an example there. Flagella is plural, flagellum is singular. In the human body, you do have cilia lining your entire respiratory system, so people who get a bad smoker's cough get that because they basically kill the cells lining their respiratory tract, and the cilia can't move and push the mucus and other trapped particles up and out of their lungs. That's normally the function of them. So since those are killed, you get a terrible cough that doesn't go away. The last thing is the cytoskeleton. So it's not just like a water balloon. A cell is more like a water balloon that has a whole bunch of toothpicks running throughout it, holding it in place, except the cell is able to break down those toothpicks on one end and build them up on another to get the cell to move on its own. That's what the cytoskeleton does. It provides support and structure for cells. The organelles get anchored in place by the cytoskeleton. There's a couple different types. There's microfilaments and there's microtubules. Microtubules are more hollow. Microfilaments are more single-stranded. Both of them do similar things, but they also help cells move. So as cytoskeleton is getting torn down in the back part of the cell, it's getting built very quickly in the front of the cell, and that pushes the cell forward. And that's how something like an amoeba or a white blood cell can actually roll along and move and stretch. All cells have a cytoskeleton. As we talked about before, all cells have a cell membrane. All cells are going to have ribosomes. They may not have their nucleus where their DNA is, but they're still making proteins even if it's a prokaryotic cell. So they all have ribosomes. They all have a cytoskeleton. They all have cytoplasm. And that's all of the cell parts. Make sure you study them, make sure you know them, and the next thing we'll learn is how they interact with one another to make the cell function.